All right. Good morning, everyone. Thank you, Dr. Mackey, for the wonderful introduction. Very happy to be here today. I'd like to take a moment to introduce you to the Pain Center at WashU. You said 20 years, and that still takes me aback a little bit. I started 20 years ago as a pain center of one. The kind of three panels to the left here, let me get my pointer going. Okay. These three groups are the folks who comprise the kind of card-carrying members of the WashU Pain Center, a group of basic scientists, clinical researchers. I just added Alex Jamessian down here at the bottom, who just joined us yesterday. And then a large clinical practice. Our clinical pain management group is a collection of various care providers, anesthesiologists, physiatrists, psychologists, and pharmacists who do a wide variety of multidisciplinary approaches to management of pain. But there's a much broader pain research community at WashU as well. I've listed a number of those folks over here. But the core missions of basic and clinical research, clinical practice, and education that we've built up over the years, and something I'm very, very lucky, fortunate to be a part of. So probably don't need to introduce pain to a group of anesthesiologists. Obviously, it's a very important sensory modality that has an important protective function, eliciting reflexive withdrawals. Pain is not a static sensory modality. It's very plastic, and so becomes sensitized even after simple injuries. And of course, the notion is that that kind of sensitization serves a protective function well to aid in wound healing, postoperative pain, for example. But then, of course, another important aspect of pain is that it serves a really important learning function. This is one of my friend's dogs here who had a close encounter with a porcupine. And I assume that the dog would not approach that animal again. Of course, as humans, we can know these things a priori. We've learned, we've been taught, and then you wonder about how this sort of thing might happen. I hope this fellow would learn from that, but maybe not. So, of course, what we're concerned about really is not these kind of acute pain conditions which serve important survival functions, but really the more debilitating chronic pain conditions, some of which I've tried to illustrate here, things like phantom limb pain, migraine headache, chronic low back pain, fibromyalgia, visceral pain syndromes, et cetera, that really don't serve any protective function and have the aforementioned really huge impact on quality of life. And these conditions are immensely difficult to treat. The Institute of Medicine report, hard to believe how long this has been ago, Sean, who was very involved in this process. But as he mentioned, indicating, somewhere in the neighborhood of 50 to 100 million people impacted by chronic pain, huge economic impact, and of course, desperately in need of new treatments, which is what we're in the business of doing. So, little introduction to what my lab does. It's kind of wide across the board. My training is, I'm a neuroscientist, a PhD neuroscientist by training. The core of what I've been studying since I started my lab was understanding mechanisms of sensitization in pain pathways, with the idea that sensitization itself is key to the kind of chronification of pain, and something that should be treated. And we'll talk about whether or not that's actually true, or whether chronic pain sensitization sort of reports on the presence of clinical pain. The lab over the years has evolved from thinking about kind of sensitization at a cellular level to really stepping back as new technologies have become available, really think of it as a problem of circuit dysfunction, and understanding how multiple levels of adaptation in pain pathways can lead to this clinical type of sensitization. And then can you think about these kind of circuits more broadly to find levers there to push and pull things to get things back to normal physiological function. So a lot of kind of systems level neuroscience work that we've done in animal models to get at that, all with the goal of identifying targets that we might manipulate to normalize these functions. Part of that circuit neuroscience approach took me down a path about 10 years ago of developing enabling technologies for studying these circuits in vivo. And that has come full circle from developing sort of tools for animal research to thinking about how you might implement technologies like optogenetics for the treatment of pain in patients, but also adapting those technologies for implantable systems for treating pain and opioid overdose, which I won't be talking about today. What I will be talking about is these sensitization mechanisms and how we can learn from animal models and then do different types of studies, either in healthy volunteers or pain patients or in human tissues, to kind of provide a first level of translation from animal models to patients. And how can we predict better how something is going to translate from these preclinical studies into new treatments. And as part of that process, the way these treatments, ideas about potential treatments become a reality is through commercialization of those things, and not with the goal of, the financial impact, but that is how a medication or a new device gets made available to patients. So we have quite a bit of involvement in that. So today I'm going to talk, like I said, about these sensitization mechanisms, how we translate them, and where we are with respect to new therapeutic development. And we'll start with kind of thinking about this problem of kind of lost in translation. Where have we gone astray in the development of new pain treatments? As I mentioned on this slide, the past 25 years, we've seen, I guess maybe I should say 30 now, incredible advances in our understanding of pain neurobiology, really a renaissance in the pain field in terms of understanding molecules that mediate transduction in the periphery, understanding this, at very fine detail, the cell types and mechanisms that mediate pain, and identified, a lot of mutations. I think has been a big advancement in the field, is identifying mutations that cause congenital insensitivity to pain and hypersensitivity, congenital pain, inherited pain syndromes. So all of that work has identified what we think are potentially useful targets for the development of new interventions for pain that could block pain either at the source, so at the nociceptive transduction mechanism, or in its transmission to the central nervous system, or even in modulation centrally. And there were a lot of investments by big pharma in developing these new things. And so the question is, where are the new treatments? And there are a few, but there are a large number of notable failures. The kind of poster child for this was NK1 antagonists very early on. But there have been a bunch along the way where animal models suggest that things like NMDA receptor antagonists, nicotinic receptor agonists, 5-HT3 antagonists, microglial activity inhibitors, all these things would have been predicted to have efficacy for treating pain, and had substantial evidence base from preclinical models, seemingly good compounds with drug-like properties, and large trials that all basically failed. And so why? There's plenty of blame to go around. At least everybody is convinced that they know why it is. But varyingly, animal models are not predictive of efficacy in human. On the one end, you messed up the trial. Why would you select that patient group for studying this? To more mundane things, but really important in terms of first validating target engagement and things like that in the clinic. So I guess what I come down to, and what I'll kind of talk about today, is a problem in pain research. And this is not an indictment of animal studies. It is just simply animals are what they are in terms of being useful for identifying mechanisms and nominating candidates that you could interrogate, but they're not little people. And so there are things that we need to do, steps that we need to take along the way. So what do I mean when I say mice are not little people? Well, pain's really hard to assess in a rodent model. Mice and rats are the predominant animals that we use for these preclinical studies. So when we do these studies, we generally ask a mouse a very different question than what we ask of a patient. We'll get into that. And what we can ask them has an unknown relationship to clinical pain. And secondly, in the most cases, the relationship between the physiology of the rodent and human is unexplored almost completely. There's, they're mammals. They have neurons that look like what we have in humans, but the question is how different might they be based on the environments that these critters have to navigate. So what we've been doing, I guess about 15 years ago, kind of started on this process to really look at that translational interface from animals to humans and to figure out what do we need to do differently. And so with those couple of previous slides in mind, we've tried to do things that allow us to align what we're doing in early human studies with what we're able to do preclinically. And that means you can adapt things on either side. And we'll get into that a little bit. And then secondly, we've spent a lot of time doing working with human tissues that we obtain from organ donors and patients to do comparative physiology and genetics to understand differences in just where these targets are expressed. And that'll be kind of the meat of what we talk about is how we've used that to inform how we should be advancing or not our candidates. Okay, first thing, pain is difficult to assess in animals. Visual analog scale, how do you feel today? Scale of 0 to 10, no pain or worst pain imaginable. And if you ask that to a mouse, you don't get a lot of an answer. So what do we do? What can we ask of a mouse? Well, we can do things to assess sensory thresholds. These little apparatus here is for, they call this the Hargreaves test. Ken Hargreaves, a well-known pain scientist, but just a way to do basically quantitative sensory testing in an animal. So this is a little heat probe that you can heat the paw of the animal and look for withdrawal response. And that can become, with nerve injury or inflammation, they become sensitized and become more reactive. Okay, so they're sensitized. On the mechanical side, we can use things that you use in patients, so von Frey filaments or other types of probes to test mechanical sensitivity. So these are easy to test. You can measure for sensitization, which is apparent. I showed that picture of just a paper cut. That hypersensitivity happens, hypersensitive to heat and touch. And then the question is, does that have anything to do with actually the kind of pain you would want to treat, ongoing kind of pain? And that's unknown. So what do we do? So we do, these sensitization experiments in animal. We test maybe a target. And if something goes forward to a clinical trial, we give them this new test medication and ask them, so how do you feel? Do you feel better? So what we need to do is something that aligns those two. Perhaps it's just that we're asking the wrong question. So we can either try to ask mice how they feel, and there are a number of ways you can do that. One of the approaches has been to use kind of pain-suppressed behaviors. So things that mice or rats would normally do, and maybe pain causes a decrease in that sort of activity of daily living, if you will. Things that people have tried, things like voluntary wheel running, gait changes, home cage activity, facial grimace even seemed absurd at the beginning, but actually seems to be something that people use quite a bit and has some validity for really strong pain conditions. So all these kinds of things basically get at these kind of reduced activities of daily living, which you see in patients with chronic pain. And so there's been a lot of work in that area with varying degrees of success. And then to do other types of more sophisticated behavioral assays where you give an animal a treatment and you get them to report back to you on how they feel by whether or not they develop develop a preference for, say, an environment where they've received a treatment that is effective. So that's how you ask mice how they feel. On the flip side, maybe we could do something seemingly more simple, at least conceptually, because it's hard to ask mice how they feel, which is before you go on to asking patients whether they feel better when you treat them, do that assessment of sensitization first in a healthy human volunteer kind of population. So kind of repeat the experiment in a person, okay? So measuring sensitization, we can do the same thing. There are lots of more expensive ways to do this with these kind of, this is the mouse apparatus. In humans, we use these kind of thermal, Peltier-controlled thermal probes that allow us to heat or cool the skin. We can assess sensitivity to heat. We can still do various forms of mechanical sensitization, very similar to what we would do in the rodents. And so now, is that a valid approach? So what is the evidence that sensitization actually happens in patients? That I'm going to show some old data that we've collected, but there's actually a large literature on this from a lot of different people. This is a study we did a long time ago now in migraine patients, just asking whether we all know that patients who have migraine, when they're having an attack, they're very sensitive to a variety of stimuli, sound, light, touch. But the question was, is there evidence that these patients show sensitization, what I'd call interictally? So when they're in this case, these patients were all 24 hours removed from a headache, either before or after, and they were headache free at this time. And this is just a study that I did with Todd Schwett when he was at WashU, but basically doing QST on the forehead or the forearm, measuring heat and cold. And you don't need to look at the details really, but both episodic migraineurs and chronic migraineurs showed hypersensitivity to heat and cold on the trigeminal innervation areas, but also on the forearm, suggesting that they have a generalized kind of hypersensitivity to at least thermal stimuli that's present indirectly. So they do show evidence of sensitization. And so might that be causal, or is it reporting on the presence of chronic pain? We're not sure. Similarly, a few years later, working with Henry Lai as a urologist at WashU, urologist surgeon, we tested patients with interstitial cystitis or bladder pain syndrome and did pressure pain thresholds on the suprapubic region of the abdomen, and they show referred hyperalgesia to the abdominal wall, which is something that we see in animal models as well. So this kind of sensitization is present. This has been seen in lots of different pain conditions. It's sort of fibromyalgia, that's a descriptor of what that is. It's lots of hypersensitivity, but you can see this in lots of different patient populations. So the general idea is, okay, sensitization is present in chronic pain patients. So instead of going from asking an animal about sensitization and then asking a person how they feel, maybe the first thing we do is we can test whether these things that reverse sensitization in animal models can do the same in human subjects, healthy human subjects, and experimentally induced sensitization models, which I'll show you one of those in a minute. And then the ones that work in that kind of first level translation, if you then test that in a chronic pain patient, in a migraine patient or a bladder pain patient, do you reduce that evident sensitization that's present there compared to placebo? And then lastly, the ones that work in that respect, by the way, how do you feel? Does it actually reduce your ongoing pain? And it's sort of an unknown relationship between that ongoing sensitization, because it's possible you could give something that would reduce the kind of sensitivity, hypersensitivity phenotype, but would not really have an impact on reported pain or activity of daily living, or just overall, how do you feel? So I'm going to tell you a couple of stories today, backing away from the general concept to what I started studying back when I was a graduate student. Metabotropic glutamate receptors is a family of G protein coupled receptors that serve neuromodulatory effects in excitatory pathways throughout the brain. And there are 8 of these in mice and rats and human. They're in these 3 different groups. There's the group 1 receptors tend to be postsynaptic excitatory receptors, so their activation might produce sensitization in a pathway. And these group 2 and 3 receptors are inhibitory, kind of presynaptic receptors that tend to dampen synaptic transmission and excitability. And so you might expect that either antagonists of these receptors or agonists or positive allosteric modulators of the GI receptors could be used to reverse sensitization, which is how I started my research lab. Having studied these for years, had a lab where I wanted to start studying sensitization in pain instead of thinking about other parts of the brain. I was just asking, what do these receptors do, and how might they be taken advantage of with the growing arena of pharmacology for these receptors to modulate pain. So I'm going to do a couple of little case studies of how we've gone through this kind of micro translation pathway. The first we'll talk about is mGluR5 induced sensitization, summarizing about 15 years worth of work that we had done and others. Basically, we found that mGluR5 is important in mediating sensitization at multiple sites throughout the pain neuroaxis, notably in the periphery, in the spinal cord, dorsal horn, and in the amygdala, in a variety of mouse and rat studies, and monkey. And secondly, mGluR5 antagonists in a lot of these animal models reduce sensitization in models of inflammatory, neuropathic, and visceral pain. So skipping all of that, just to say there was a substantial kind of preclinical evidence base. And then the question is, does that translate to humans? So with all that in mind, we went to the books, looked it up to go through all the FDA-approved drugs that have mGluR5 antagonist activity. This is an exhaustive list right here. So we had to go starting at this point. I knew that there were programs in development, but we needed something that would allow us to kind of try this first level test in humans and then potentially first validate in mice. So frustrated by the lack of an available tool, kind of waiting. And then there was a report in 2005, way back when, by folks at Hoffman Roche about this compound phenabam. Phenabam was originally developed in the 1970s by McNeil Labs as a non-benzo anxiolytic. Back then, that kind of drug discovery was you have chemical libraries that you inject into rats and see if they have reduced anxiety-like behavior. And then if you have a positive hit, you try it in people. It's very cavalier, but no mechanism of action. But very effective in rats. It was actually tested in humans. It was found to be safe and effective in the treatment of severe anxiety in patients in a trial in 1982, very quickly moving from the lab to the clinic. But then it had kind of wonky pharmacokinetics, and so it was a very difficult compound to work with. They went back to try to make modifications to the molecule to fix that, but when you don't know what the target is and you can't screen it, you lose your way pretty quickly. So they kind of got mothballed. But in the early 2000s, there was a lot of work on screening for new antagonists, and what they have libraries of compounds that have been tested in people, and they would be screened through as part of a process to identify antagonists for the receptors. At this point, the metabotropic glutamate receptors had all been cloned and identified, and this popped up on a high throughput screen by Hoffman Roche and showed to be an antagonist or a negative allosteric modulator for mGluR5. And so this was my little gift from the science gods, which is an mGluR5 antagonist that had patient safety data, and very simple, right? I just need to get some of that compound and test it in people. It doesn't work like that. But anyway, first due diligence. So let's test it in mice. Go back to the mice. We did a bunch of stuff to just say, does this drug actually work to relieve pain in mice? We repeated a bunch of stuff that had been done. This is the formalin test. It's just injecting an irritant into the paw. There's spontaneous pain behavior that has a couple of phases here. And you can see treated with vehicle or treated with phenabam, you get a nice suppression of this kind of irritant-induced pain. To show that this was on target, we used mGluR5 knockout mice, so they just lack the receptor completely. There's reduced pain behavior in those mice, but the residual pain behavior is no longer suppressed by phenabam. So that's a good sign that the drug is working through the proposed mechanism. We repeated in a couple of different models. Details are important. This is an inflammatory pain model using that heat sensitization model. It was very effective there. We used a bladder pain model, distension of the bladder that produces a reflexive pain response in mice and rats. And we get a nice, this is increasing pressure in the bladder, and the pain response scored through a visceromotor response here. And you can see a nice suppression of that behavior there. So the last question, ask the mice how they feel. So the way we did this, we didn't do this. This was by Ben Kolber, who is a former postdoc in the lab, but he did this in a nerve injury model. So the notion is, so we've seen all these kind of evoked pain measures. So in trying to ask the mice how they feel, this is animals that all have, this group has nerve injury, spared nerve injury. It's an injury to the sciatic nerve that produces kind of a traumatic neuropathy. And those animals show robust sensitization to touch and cold, or sham operated animals over here. And what we're doing here is a conditioned place preference assay. So you put the mice in a chamber that has certain visual or olfactory cues versus the other chamber, and you pair one chamber with the drug and one chamber with a placebo, and you do that a couple times, and then you put them back in the chamber a couple days later, and they could float back and forth between, and where they spend their time, they're telling you where they prefer. And so the notion is, if you have something that is analgesic, it will produce a place preference in an animal with pain. And if it is not rewarding but analgesic, it will only do that in animals with pain, and that's what you see here. So the phenabam animals show a preference in animals that have nerve injury, but not sham-treated animals. If you give something like morphine, you'll get preference in both cases because there's rewarding features of the drug. So the good news is, asking mice how they feel, they tend to like this, but they only like it when there's pain. And the interpretation of this in the field generally is that's preference due to relief of ongoing pain, such as it is. So The drug's effective in a lot of preclinical models, reflexive and non-reflexive endpoints. It acts specifically through this receptor. We didn't see any adverse effects. We did a bunch of liver tox and blood chemistry and all this kind of stuff to see if there's any detectable side effects. The only thing you get is locomotor stimulation, which is not really a big problem. And in human trials, there have been mostly a lack of serious adverse events, and it has been shown to be effective in the treatment of severe anxiety in patients. So does this work in people? So here we go. Very simple, just do a study. And so this was my education about how complicated it is to do these kinds of things. It took us, because of the state of the intellectual property around this, it was very complicated to get an IND to do this. So we had to go through the process of doing GMP synthesis for use in human studies. Getting the IND was challenging. We had incredible partnership by folks at Janssen, J&J, who pulled the records out of mothballs that we needed to have the data for the IND. Because remember, this was from the 1970s. And so I'm envisioning, some sort of warehouse Raiders of the Lost Ark situation where they could go and find a banker's box that had files in it. And I got this CD with all the scanned data that allowed us to get the IND. So many thanks to them. It took 6 years to get to that point. Enter my colleague, Laura Cavallone, who was an anesthesiologist working with me at the time, and she helped to design a study to get at this. First thing we did was to do some pharmacokinetics after oral dosing with phenibam. And these are 3 different doses of phenibam. You can see a nice dose-response relationship here in terms of plasma concentration of the drug over time, peak at about 3 hours after oral dosing. You'll notice the lack of error bars here. The reason for that is it's a mess. As I said before, the original study showed that there was really widely variable exposure, which is definitely true and creates a problem. But overall, there's a dose effect. The question is how many patients, how many subjects? No adverse events that were reported. And Sean, we were talking about this yesterday. We chose to use the heat capsaicin sensitization model, which is a model for inducing central sensitization in human subjects. You basically put some capsaicin cream on an area of the forearm, and then you heat the skin in that area with the thermode. You can see this is my arm here. So there's the outline of the area. You can see the flare here. And then you can measure with foam brush or Von Frey hairs outside of that. You get an expanded area of sensitization, which is believed to reflect the development of central sensitization. And it's been used you can demonstrate efficacy of morphine, cannabinoids, gabapentin, and others to reverse that sensitization. So kind of validating it somewhat as a preclinical model for this. So we did this model in healthy subjects and measured this area of sensitization. So this is kind of that area of sensitization on the forearm over time. And you can see that this kind of wanes over the course of a couple hours. And these are placebo-treated folks, and these are subjects that were treated with phenibam. And the drug was given 3 hours prior to inducing sensitization. We did have a significant reduction of sensitization that happened in a small, healthy volunteer cohort at the beginning. So it was very messy because of the variable pharmacokinetics that I mentioned. But nonetheless, I felt was a good window into the notion that at least the sensitization that we saw in animals has a similar effect in patients. So completed these kind of healthy volunteer studies. We had all kinds of questions about exposure and do subjects that have higher levels have more pain relief, but we didn't, that's not the way it was designed. So we wasn't powered for that. Maybe we needed to look at PET studies to see receptor occupancy and correlate that. So that all of that could have been done. It would have been a massive, expensive trial, and the drug is terrible in terms of potential real world application. So basically awaiting a better drug to move into clinical trials. So we'll come back to that in a minute. But that's where we were with that. We kind of did that first level of translation. The next thing would be to test it. If you had something where you had good exposure and all that, test it in chronic pain patients to see if that sensitization is gone, and then how do they feel. So that's the first part, is aligning endpoints in animal models and people, and that kind of story still points to mGluR5 antagonists as being potentially useful. The next thing that I mentioned as a tool here would be to understand the comparative physiology between rodents and humans to predict whether something is likely to be translatable from the animal model to people or not. This was all started, a dozen years ago or so. I ran into a good friend of mine. We were postdocs at the same time, a guy named Andre Getty, who had started this company, Anabios, after we'd parted ways. And his company's idea is that to redefine first in human from doing safety studies in people to first doing studies in human tissues. And so they had a network of organ procurement organizations that would work with to collect things like cardiac tissue, liver, peripheral nerves, skin, to do kind of toxicity and exposure studies in human tissues as the first estimate of whether something's going to be effective. So instead of doing a screen of a bunch of ion channels, hERG channels, et cetera, to predict cardiac toxicity, you do contractility studies on human cardiac muscle and just see, does the drug have an effect at all? So kind of speeds that up. But one of the things they had started doing was they had gotten interest from companies who were working on pain about acquiring dorsal root ganglia and spinal cord tissue for these kind of comparative studies. And so Andre and I partnered early on to use the human dorsal root ganglion as a platform for doing this kind of first in human translation. And what we wanted to do is see, can we get these out and use them for things like genetics and physiology? And working with Anabios, we got dorsal ganglia from organ donors and dissociated them, put them in culture, and we could do things like calcium imaging here. You can expose them to things that cause pain and itch, and you get nice responses of these neurons. So that kind of translates well. You can do electrophysiology, look at excitability and changes in that over time. And they work great for that. You can do things like expose them to inflammatory mediators. Here we did things like bradykinin and prostaglandin E2, known pain mediators that cause sensitization. You can get robust firing induced in these human sensory neurons by these agents. And we really became committed to the use of these tissues as a way to kind of do validation of our targets from animal studies. So we can acquire these from organ donors. We did this initially with Anabios. It's been replicated at our site. We've now helped to kind of spread this to other locations as well. You get these dorsal root ganglia, you can use them for genetic studies, so RNA-seq and in situ hybridization, immunohistochemistry, imaging, electrophysiology, all that to study whether these targets that you've studied in animal models are even expressed in the same cells and whether they have similar physiological functions in humans as that new kind of first in human aspect. So then the question is, do novel targets that you've identified in animal models translate to human cells at the same level? And so we talked about mGluR5 before. One of the places that we were studying mGluR5 from rodent models is we found it to be expressed in the periphery. And so now what I'm just going to show you is generally, let's first ask whether the receptors are expressed in the same place, same cell types in humans as are in rodents, and then do you have similar physiological effects? So part of that mGluR5 story I showed you before, we had very early on shown in collaboration with Sue Carlton at UTMB, mGluR5 expression. This is in the mouse skin. This is a REMAC bundle, C fibers at the dermal epidermal junction. And the black staining that you see there is immunoreactivity to mGluR5 out in the periphery, which was a shocker to me. Why would it be there? We won't talk about that, but basically we found that if you activate that receptor, you get hypersensitivity to heat and touch. You can drill down to a molecular mechanism, and we found that mGluR5 causes sensitization of TRPV1, which is the capsaicin receptor, which is a noxious heat transduction ion channel in the periphery. And we did a bunch of work to map the whole signal transduction pathway, receptor, and what signaling mediators are there, what kinases. We identified phosphorylation sites on the channel. So any of these things in this little complicated pathway here, in theory, should be targetable for the treatment of pain. But I was particularly keen on this peripherally expressed mGluR5, and this preceded the whole Phenibam study I just told you about. But I was intrigued by this notion because if we could target peripherally expressed mGluR5, we might prevent any CNS-related side effects, and we could target that. So with our new dorsal root ganglia in hand, one of the first things we started doing was bulk RNA sequencing on the ganglia. Actually, the first thing we did, to be honest, was we tried to do immunostaining in human DRG. That didn't work. It's just a mess. Human DRG have all kinds of autofluorescence. It's very hard to do. So we tried to do RNA, in situ hybridization and or even PCR-based amplification. We couldn't really see mGluR5, couldn't figure out what's going on. We went back and tried bulk RNA sequencing, comparing mouse to human. So this graph I'm showing you here is just the abundance of mRNA in this is mouse in blue here, dorsal root ganglion. These are all the metabotropic glutamate receptor subtypes, and you can see in mouse, mGluR1, 2, 3, 4, 5, 7, and 8 are all expressed to some extent. mGluR6 is a retina-specific gene in mice, and so it's not expressed. So we did this bulk sequencing in mouse, and then we repeated it in human DRG, and much to my chagrin, although mGluR5 is expressed at low levels, here you can see in mice, it is not detectable at all in human. And everything that we had done suggested that this is a species difference, that there's just not mGluR5 expressed there. Same thing with mGluR1. So all of that work, 15 years of NIH funding, we had started development of peripherally restricted mGluR5 antagonists, and it was just doomed to fail. I wish I had done this experiment 12 years before that, but nonetheless, it kind of informed us and said, well, okay, we're not going to work on these peripherally restricted mGluR5 antagonists. All right, so pull up stakes, but note that mGluR4, 7, and 8 are robustly expressed here. How are we doing on time? I feel like we're out of time.
16 minutes.
Okay. Oh, my timer on here, this was up before I started. I was like, wow, I really got to the end quickly. I know that nobody cares if the ORs start late, so let's just hang out for a while. We'll just do that. Right? That's the way it is at WashU. I don't know how it works here. So bummed about mGluR5, got pretty excited about mGluR4, 7, and 8, which are, note this is a log scale. So there's much more robust expression both in rodents and it's retained in humans, at least at this level of RNA-seq data. I'm going to skip over, but we have a whole other similar story about mGluR2, GRIM2. What you can see is more robust expression here for mGluR2 than mGluR5 in mouse, but in humans, it's expressed at quite low levels, but it's still present. Just all that to say, we did a whole other series of studies about mGluR2, which is a GI-coupled receptor agonist, and rodents showed nice analgesic effects. There's still a little bit of mGluR2 there. We showed some expression in humans, effects on physiology. I'm skipping this because it's sort of like there's an effect there, but it's sort of marginal, and there's some differences in the effects in human DRG neurons at the functional level compared to what we saw in mice that suggests it might still have efficacy, but it's going to be somewhat limited. So both of these kind of womp moment kind of made me think, okay, we need to pull up stakes. But I got excited about those other receptors that are so robustly expressed. I'm not going to talk about that today, but that's where we've kind of landed. So generally, from the comparative physiology part, this is just DRGs that I'm talking about. You can use these human DRG to do all kinds of studies that can really help you validate the presence of a target. We found that mGluR5 is something that should not be prioritized as a peripheral target, but given our experience in parallel with the mGluR5 antagonist in people, it basically just says that effect that we're studying there needs to be targeting central nervous system instead of the periphery, unless you imagine that there's mGluR5 in some non-neuronal cell that's influencing this. There's not really evidence of that except in paraneoplastic syndromes. But in kind of general pain, we think you'll need to target central mGluR5. I'm still excited about those group 3 mGluRs. It's something we continue to work on. So all of that work on comparative physiology has led us down that road to let's look at central mGluR5. The HEAL initiative has offered us incredible opportunities now to go back to the drawing board on things where there's not a lot of industry interest right now, but where there's good mechanistic insight. We partnered, this was a very exciting moment for me because based on those data, and we said we need to look at centrally active mGluR5 antagonists, I got to work with Jeff Kahn, who was actually my PhD mentor, who left academia, went to Merck for a few years, and then came back and started the Vanderbilt Center for Neuroscience Drug Discovery. And they continued to work on metabotropic glutamate receptors. And they had a long experience of developing various chemical series for mGluR5 negative allosteric modulators that had given insight as to why other mGluR5 antagonists had failed in the clinic. They had gone down the road for clinical development of a drug called allglutamate, which was all the way to IND-enabling studies. And the very last one they did was a long-term study in non-human primates that showed a species-specific anemia induced by that drug. It's not target-based, it's chemistry-based. So it just has to do with a very specific metabolite. So the point was they were at this situation, their program had run out of funding for that, but there was this kind of lead molecule that they needed to fix one chemical liability that would allow us to get to something we could test in humans. And so the HEAL initiative is all about where's there a good evidence base? Where do you have ideas that can be translated into a new drug, potential drug that can be tested in humans pretty quickly? And so we have a HEAL initiative funded program with Jerry Brook, Jeff Kahn, Craig Lindsley at Vanderbilt to develop next generation of allglutamate as a first-in-class mGluR5 negative allosteric modulator for the treatment of pain. The goal to eliminate that specific metabolic liability and improve the pharmacokinetics. So as part of that, we did a bunch of work in switching over to rats as our species preferred for kind of drug development in IND-enabling studies. Formalin test, again, that we did with, this is allglutamate. I'm about showing you the drug that is our clinical candidate, but we are to that point. We get nice dose-dependent analgesia. We get, this is a PET imaging study in the brain showing dose-dependent receptor occupancy. Decreasing signal here shows increasing competition for the PET ligand, so occupying the central mGluR5 at doses that correspond to the analgesic efficacy that you see in the animals, which is a nice test. We've done a bunch of work. One of the other things that you need to do for any kind of these new drugs that come through HEAL is to assess for addiction liability, rewarding properties of the drug. We do IV self-administration studies. The right panel here is fentanyl, and you can see in red that the animals escalate dosing for fentanyl. Here's fentanyl. The problem with this drug is the formulation is bad. So there's a little bit of an aversive feature of the vehicle. So these animals don't administer as much fentanyl, but they do self-administer, but they don't at all self-administer this drug. So this class of drugs shows good analgesic efficacy in rats and shows no evident abuse liability at this point. So where we are with this program, starting with that compound, we've now moved on whole new chemical series where we've eliminated that AO metabolic liability. We have sensitized the API for GLB tox studies. We've done a bunch of rat studies, but a lot of that work is ongoing now with our clinical candidate through the HEAL initiative, PSPP, the preclinical screening platform for pain. And so we are hopefully months away from a phase 0, phase 1 study to test this drug in humans. Again, informed by what we did in rats and human tissues to focus on these centrally penetrant drugs. So I never want my thing to come across as saying we shouldn't be doing animal studies, because the animal studies all pointed us towards this and it allowed us to a testing platform to say, is this even possible? But I think it's really important to think about the ways that mice are not fuzzy little people. And what do we need to do differently to assess that? Ask mice how they feel, maybe do something different at the first level of translation to people, and confirm target expression in human tissues wherever possible. I think these human tissue studies are really invaluable and a much needed resource for the whole field. To, like, if there was just a database I could have gone to look at expression, I could have known right away not to really focus on that peripheral mGluR5. And I think we're at a place now where certainly centrally active mGluR5 antagonists are something worth considering, worth trying. I'm very excited to hopefully we'll be able to have that as an actual shot on goal in the next couple of years. And I'm very excited about these group 3 mGluRs based on both animal studies, but the robust conservation of that expression in humans. So kind of my new model to think, so this is the old view about how we might do translational research. Identify a mechanism in a rat and develop something and ask people how they feel. We've modified that significantly using confirmation of expression in human tissues, modification of what we're testing in animal models, relying a lot on trying to use human tissues as a starting point for identifying candidate mechanisms through clinical samples. But again, confirming these things through using animal models as a test bed to confirm some of this, and then moving forward to therapeutic development from there. A lot of work on the human tissue front, funded first through a partnership with Anabios and then by NIH. The HEAL initiative started a program called the Precision Human Network, the goal of which is to provide that big database I told you about. So we're one of 4 sites, us, UT Dallas, Harvard, and Penn, that are charged with developing atlases of expression of human pain genes and proteins in DRG, spinal cord, parts of the brain, trigeminal ganglia. This is our local site. I knew I had to come up with an acronym if we were doing something with humans. So INTERCEPT is our Integrated Research Center for Human Pain Tissues. The teams, these are all folks at WashU, Jeff Milbrand, Aaron D'Antona, Peter Jen. They're doing atlases of peripheral nerves. They've done this in mouse, and we're doing this in human, looking at healthy peripheral nerve, injured peripheral and painful peripheral nerve, and neuromas to understand expression of what changes in human peripheral nerves and what's different from the animal models. Valeria Cavalli and Goyan Zhao are doing spatial proteomics on human dorsal root ganglia and comparing and mapping that onto transcriptomics to understand the relationship between protein expression and gene expression there. And myself with Brian Kopitz, we're doing functional characterization of human DRG neurons and spinal cord dorsal horn neurons from organ donors, and doing what's called patch-seq. So you can record electrical properties of the neurons, see how that is different in patients with a history of chronic pain or opioid exposure, understand what gene expression has changed and how that maps onto functional types, and then mapping dorsal horn neurons in the spinal cord to see how well they align. All this is to become available as public data sets so people cannot spend 15 years studying something that's not expressed in humans. So, although mice are not little people, I think they're important, but we shouldn't pretend that they are little people, and just be aware. So with that, I'll thank you. I want to thank all the folks in my lab over the years who have worked on all this. Really want to thank Anabios and Mid-America Transplant, who's our local organ procurement organization who partners with us on this. My colleagues at Dallas, Ted Price, who's been my partner in crime with this for a while, Karen Peterson, Mike Robotham at UCSF, who helped me get the capsaicin sensitization study going years ago, and my folks at Vanderbilt on the project there. And of course, funding from the NIH HEAL initiative and other locations for supporting this over 20 years, as Sean said. So thank you. I'll take questions.